Hello everyone, welcome to the last eye video. In this video I'm going to highlight the path of light through the eye and just finish talking about some of the functions of the eye or how the different anatomical structures work. So this is an unlabeled image of the eye. Hopefully you're familiar with the basic anatomy. I've chosen an unlabeled image just to keep things uncluttered. And I'm going to outline the path that light takes as it moves through the eye. So you have light waves coming from an external object that you're looking at, right? And as those light waves approach the eye, the first thing they pass through is the cornea. As they pass through the cornea is curved. It does act as a bit of a fixed lens. What I mean by a fixed lens is that it bends the light, but adjustments can't be made. Like you can't change the curvature of the cornea. So it always bends the light in the same way. The light that passes through the cornea will then enter the anterior cavity, and some of that light will hit the iris, and that light is obstructed. It does not progress any further. It does not enter your eye. But some light will pass through the pupil, right? And the light that passes through the pupil will hit the lens, and then the lens is an adjustable lens. It can change its shape to cause the light to bend further. And so you will have the light pass through the lens, and then that light comes like this. The light that passes perfectly through the center goes straight. This light kind of crosses over like that. Okay? And so from there, the light passes through the lens, through the posterior cavity and the clear jelly-like fluid that's in it, known as the aqueous humor. And then that light hits the retina. And imagine you were looking at a person, right? Because the light waves cross one another, the image of that person is actually projected on your retina upside down, right? Notice that this right there is the fovea, so this is the area of sharpest vision, right? From there, what happens is that the rods and cones are stimulated. They initiate action potentials on the axons, and the axons carry those electrical impulses through the optic disc into the optic nerve, which then eventually leads to the occipital lobe of your brain. So to recap the path of light through the eye, it goes through the cornea, through the aqueous humor of the anterior cavity, right? Through the pupil, through the lens, through the vitreous humor of the posterior cavity to the retina, right? And that's where the path of light stops, right? The lens is what focuses the light, so you can focus on objects that are different distances away. I'll get into how that happens, or I'll recap how that happens in just a little bit. But let's go ahead and just finish this off by talking about the flow of the sensory information, right? What happens is that the light stimulates photoreceptors, right? Cells that are sensitive to light energy. Those are the rods, which give you black and white, or what you can think of as grayscale vision. If you're familiar with black and white film, or say an old-fashioned black and white TV, how it gives you vision in grayscale, that's what the rods do. The rods can detect a light, but they detect all wavelengths of light. Because they detect all wavelengths of light, they can't discriminate between different colors. But they're very light sensitive, and this is what's responsible for enabling you to see in low light visions or low light situations. you'll notice that in low light situations, your ability to discriminate color is diminished. Now, the cones are the photoreceptors that perceive color. There's 3 different populations of cones. One detects red light, the other detects green light, the other detects blue light. Kind of like an old-fashioned TV, emits light in those 3 colors. And what happens is that the combination of red, green, and blue cones that are stimulated is then kind of pieced together in your brain to put together the various colors. But to get back to the pathway, the rods and cones are stimulated, and then that stimulates the neurons of the optic nerve, right? And then the optic nerve follows it through that pathway discussed in the cranial nerve lecture slide, eventually to the occipital lobe, right, where it goes to the primary visual cortex. Now, let's talk about the pupil, right? So the pupil has 2 layers of muscles. There's an outer layer of muscle that you can see here that's arranged radially, right? That's called the dilator muscle. You might remember that when a radially arranged muscle contracts, it pulls away from the center, right? Because of this, when your pupil dilates, right? When your pupil gets bigger, that's largely due to the constriction of these radially arranged dilator pupillae muscles. Right? Now, there's an inner set of muscles on the iris that is circularly arranged, right? And when circularly arranged muscle cells contract, they squeeze inwards. Remember, you have circularly arranged muscles, and when they contract, they pull inwards. And this causes constriction of the pupil, right? The pupil is getting smaller, right? When the pupil is small, less light enters. When the pupil is dilated, more light enters. And so under high or bright light situations, the pupil contracts to prevent too much light from entering. It also constricts when you need to focus on objects that are very close up. Or under low light situations, the pupil dilates to allow the maximum amount of light to come in, or at least enough light to come in so that it sufficiently stimulates the photoreceptors. Okay. Let's go ahead and talk a little bit more about the relationship between the lens, the ciliary body, and the suspensory ligaments. Okay. So I'm going to try to recreate this from an anterior perspective. In black, I'm drawing the circularly arranged ciliary muscles. Right. Oops. Sorry. Right. In red, I am going to draw the lens. I'll just abbreviate it L. And then in purple, I'm going to draw the suspensory ligaments. Now, this is the arrangement if you look at the eye from the front. And so when the ciliary muscle is relaxed, there is tension on the suspensory ligaments, and the lens is pulled into its flattest position. Try to keep the colors consistent. The lens is pulled into its flattest position. That's a little bit of an exaggeration. And there's the suspensory ligaments, right? And there's the muscle of the ciliary muscle. Now, when the ciliary muscle relaxes, what's going to happen, when the ciliary muscle contracts, because the ciliary muscle is circularly arranged, it's going to close in on itself. The ciliary muscle is going to get closer to the lens. what that's going to do is that's going to release tension on the suspensory ligaments, and the lens will be able to assume a more spherical shape, which is its natural inclination. Right? This is part of the process of focusing light, which is also known as accommodation. Right? Right? And so we have contracted ciliary muscle, and we have relaxed ciliary muscle. By default, your ciliary muscle is typically relaxed, which pulls your lens into a flattened shape, which allows you to naturally focus on objects that are further than 20 feet away from you. When you need to focus on an object that's closer than 20 feet away, roughly speaking, what happens is the lens, the curvature of the lens needs to be progressively more and more increased the closer the object you want to focus on is. And so to focus on objects that are close up, the ciliary muscles need to contract to allow the lens to become more curved. So that's a little bit more than I would normally cover in a lab, but we won't be covering the function of the functional anatomy of the eye in the lecture portion. So I just wanted to give you the background that you'll be expected to have when you move on to AP 151.